Good day to all the assessor. My name is Win and my topic today is Parkinson disease. It is a very common neurodegenerative disease affecting wide populations of elderly globally, and its manifestation can be divided into motor and non-motor. The motor symptoms are like dyskinesia, movement stiffness, and resting tremors, whereas the non-motor features including constipation, emotional disturbances, and cognitive deterioration. All these will gradually worsen to ultimately loss of total independence as the disease progresses. Parkinson disease can be defined as a selective degeneration of the dopaminergic neurons in the substantia nigra that leads to a reduce in dopamine levels in the caudate nucleus and putamen, and the accumulation of intracellular protein aggregate known as the Lewy bodies, which is composed of misfolded alpha-synuclein. This figure shows the current pharmacotherapy drugs mechanism and side effects for the management of PD. They provide symptomatic relief with levodopa remaining the gold standard for motor symptoms management, whereby they increase the dopamine level in neurons at the substantia nigra. However, current therapy have on and off phases. The on phases is when the drug is working well, symptoms are controlled. The off phases is when the concentration is wearing off, so the medication is no longer working and the motor symptoms return. However, none of them provide a long-term solution, as when the neurodegenerations progresses, it deteriorates, and then the off phases will eventually get longer. So because it doesn't address the underlying neurodegenerative process. Hence, the aim is actually to develop an effective disease-modifying treatment. So in order to target those the neurodegenerative pathogenesis mechanism to slow down or to halt or to reverse the neurodegenerative cascade in PD to further sustain or enhance patient quality of life. Now let's go in depth into the pathogenesis of PD. SNCA mutated gene is one of the pathogenesis, and it encodes the alpha-synuclein proteins, and then it's found to associate with an increase of the SNCA mRNA level, and hence also an increase of production of the alpha-synuclein level in PD cases. The third mutated gene found in PD is GBA1 gene, which encodes glucose cerebrosidase 1, short form GCase1. It breaks down the glucosylceramide to glucose and ceramide in lysosome. So a decrease in this GCase1 activity also causes a dysfunctional lysosomal activity, and again increase the alpha-synuclein accumulation. In the nature, alpha-synuclein is actually a helically folded tetramer, but when there is accumulation of the neuronal glycosphingolipids caused by the deficiency GBA1 protein, it reduces the normal level of tetramers but increases the pathogenic alpha-synuclein monomers level, and hence the formation of pathologic alpha-synuclein fibrils damaging the dopaminergic neurons. Mitochondrial defect is also one of the hallmark in PD, when there is altered mitochondrial structure and dynamics, which leads to an increase of the reactive oxygen species, and hence also disrupt calcium homeostasis and decrease production of ATP. So this one actually causes more neuronal cell damage, as dopaminergic actually requires enormous mitochondrial energy supplies for normal physiological function. The pathologic monomeric alpha-synuclein will aggregate to form amyloid fibrils, and the fibrils will continue to aggregate in Lewy bodies. These amyloid fibrils induce neuroinflammation by interacting with microglia toll-like receptor 2, short form TLR2, and NLRP3 inflammation, in which stimulates nuclear factor kappa B pathway. This activates the microglia to release TNF alpha and interleukin-1 beta. The function of insulin includes transporting the glucose through the blood-brain barrier to provide energy supply to neurons. There are two main pathways to ensure survival and to prevent apoptosis of neuronal cells in brain tissues, which are P13K-AKT pathway and UPS pathway. However, insulin resistance causes dysregulation of both of these pathways, hence causing neurodegenerations in Parkinson's disease. Now I will go to potential targets to develop a disease-modifying therapy. Alpha-synuclein would definitely be one of the targets, as it's a main hallmark in Parkinson's disease. There are several methods in development, including vaccinations, immunizations, and so on listed here. For monoclonal antibody, it actually developed to aim different site of alpha-synuclein protein to promote immune-mediated clearance of the pathological alpha-synuclein and enhance hope to reduce cellular toxicity and interrupt the prion light spreading mechanism. Monoclonal antibodies in development includes prasinezumab, has already gone through phase 2 trials. For patient receive a dose of 1,500 mg for 5 years. There is a slower motor symptoms deterioration at year 4 and also an improvement of the Unified Parkinson Disease Rating Scale of 118% during the on phases. Alpha-synuclein vaccine, as the name implies, it induces the endogenous antibody production to inhibit or clear alpha-synuclein aggregation. PD03A vaccines has shown to induce antibodies productions against pathological alpha-synuclein aggregates with improved immune response intensity and specificity in phase 1 clinical trial, and also proven to be safe and well tolerated. However, there's no improvement in PD motor and non-motor symptoms observed yet, so more studies are still required. Small molecule inhibitors prevent fibril formation by stabilizing the physiological alpha-synuclein conformations or disrupt pathological aggregation pathway. So there are two in current trials. NL138B in phase 1 got good safety profile but still need more studies to explore its effectiveness. And minzazolmin has shown robust effect in mouse model, but unfortunately lack of efficacy in primary endpoint in phase 2 trials. It's suspected that maybe the drug concentration is still inadequate to deposit in specific region, which is especially the substantia nigra, for clinical application. Antisense oligonucleotides and small interfering RNAs can selectively decrease SNCA mRNA level, hence reduce the alpha-synuclein protein production. The other end, CRISPR-Cas9 genes editing offers the potential to permanently reduce the expression of SNCA by deleting the alleles and or amending the mutation site in the gene. The genetic approaches have a crucial challenge to overcome, which is they have to find how to penetrate the blood-brain barrier to deliver an efficiency and long-term effect. So researchers have actually tried intracerebroventricular and intranasal of the antisense oligonucleotides for 28 days into a mouse and monkey model. There is improved dopamine neurotransmission and also a reduced alpha-synuclein level in the midbrain. Whereas for small interference RNA, they use the nanoparticles to help transport to go through the blood-brain barrier in a mice model. There is a decrease of SNCA mRNA expression in the striatum of 65%, also followed by a decrease of the SNCA protein by 50%. CRISPR-Cas9, they also used to delete the A53T mutated SNCA and transported using the adeno-associated virus in the PD rat model. There is also significant reduction in expression of the alpha-synuclein and reactive microgliosis, subsequently preventing also the neurodegeneration and improved motor symptoms. But all these are still in vivo studies. To address the deficiency of GCase enzyme, there's several methods, includes enzyme replacement, small molecule chaperone to stabilize mutate GCase, and so on. Ambroxol is actually a mucolytic drug, but is repurposed to be a GCase chaperone as it can penetrate the blood-brain barrier and hence increases the GCase activity in the brain and also shown acceptable safety in early clinical trials. And another allosteric GCase activator known as LTI-291, it enhances the enzyme activity without even requiring a chaperone function. GBA1 gene therapy using adeno-associated virus vector can also deliver a functional GBA1 to the brain. GLP-1 receptor agonist is marketed as an anti-diabetic agent. It also has been repurposed in PD as it's shown a neuroprotection mechanism in PD. The activation of these GLP-1 receptors reduces the microglia activations and pro-inflammatory cytokines production such as TNF-alpha and interleukin-1 beta, potentially breaking the cycle of chronic neuroinflammations. Besides, the agonist also enhances the insulin signaling in the brain, which may improve neuronal energy metabolism and survival. Currently, few GLP-1 receptor agonists has been evaluated in PD clinical trials, including asinotide, lisasinotide, and liraglutide. Mitochondrial dysfunction is also a well-established feature of PD pathogenesis, supported by genetic evidence and also toxin model using mitochondrial inhibitors. This has motivated the development of therapies aimed at enhancing the mitochondrial function, reducing the oxidative stress, and promoting the mitochondrial quality control. Most recent mitochondrial repair approaches includes biosynthetic precursor of nicotinamide adenine dinucleotide NAD+, such as the nicotinamide riboside via oral administration, shown to increase the cerebral NAD+ level and enhance the mitochondrial metabolism and cellular energy production. As PD specifically involves dopaminergic neurons degeneration in the substantia nigra, stem cell-based therapy technically should work even with just a transplantation of a single cell type. Modern cell-based strategies includes human embryonic stem cells or induced pluripotent stem cells differentiated into the dopaminergic neurons. The first clinical case of induced pluripotent stem cells transplant surgeries on the PD patient is reported in year 2020. The transplant was carried out both left and right putamen with a 6-month gap in between. The outcomes are very enlightening as the graft survived and the patient also has improved clinical symptoms from 18 to 24 months post-surgery and not requiring any immunosuppressive drugs. Parkinson's disease is a syndrome with multiple subtypes that have distinct underlying pathologies and progression rates. All this contributes to the heterogeneity of the disease and hence complicate clinical trial designs as therapeutic effect may be diluted in heterogeneous population and subgroup analyses are often underpowered. So the field is moving toward a more precise patient's PD subtype stratification based on genetics, biomarkers, and clinical phenotypes. However, validated stratification approaches are still emerging. Hence, precision medicine approach targeting therapies to patients most likely to benefit from the said depends on what subtype the PD is. They use appropriate disease-modifying therapies as a target to that pathogenesis pathway will benefit. This represents a major strategic shift. Rational combination also seems meaningful as they target different pathogenic pathway, including a combination of alpha-synuclein targeting agent with a GLP-1 receptor agonist or a genetic modulator with an anti-inflammatory agent. However, combination trials are complex and expensive, requiring careful consideration of safety, pharmacokinetic, and trial design. Disease-modifying therapy development for Parkinson's disease is now the main focus, but there are still challenges to overcome in few future decades. While there's no DMT yet to be approved use for PD, the breadth and depth of current research effort combined with improved understanding of the pathogenesis and more detailed trial design, meaningful progress is achievable to transform this therapeutic landscape of PD disease. These are my references. Thank you for your kind attention.